the universe. This is JC on a bike. My name is JC, and this is Journal Club on a bike. Last day before Christmas vacation. On my way home. And I got a great short Journal Club for you. A really exciting result out of a cancer laboratory, actually, at Baylor University in Texas, I believe. And this lab has a really exciting result. And it stems from the idea that it is known that mitochondria in neurons, if they are dysregulated or misfunction, can affect the way that neurons function, can affect the way that circuits function. Indeed, there are many thoughts about the ideas that mitochondrial dysfunction could be involved in neuropsychiatric disorders like schizophrenia or any of these things. Now, specifically, this lab is looking at a chemotherapy agent, a chemotherapeutic, and I don't remember the name of it off the top of my head. It starts with a C. It's like cisplatin or something like that. And this drug, when given to patients and when applied in vivo and in vitro, can cause mitochondrial dysfunction in neurons. And the mitochondrial function in neurons is well understood, and it's thought to underlie a sort of cognitive deficit that can result from chemotherapy. So their idea is pretty simple, right? I mean, we have lots of well understood culture systems where we can culture neurons and we can co-culture neurons with astrocytes. And so what they want to do is use a culture as an investigative tool to find out what is happening with this chemotherapeutic agent. And so in figure 1, they're just looking at co-cultures, and they look at how neuronal survival changes when they apply this cisplatin, it's called. And when they apply cisplatin, what they see is that neurons don't survive as well. And then what they notice is that if they co-culture neurons with astrocytes, then their survival rate goes up. And that's just the basic observation that the paper starts with to show you that this co-culture versus neuronal culture, pure neuronal culture, there's a difference in terms of the way that cisplatin affects survival in this prep. So in the next figure, what they're going to do is look at mitochondrial activity using fluorescence, and it's not immediately clear to me exactly how they do this, but what they do is they look at calcium dynamics. And they can see that at the resting membrane potential, the level of calcium in the neuron is already elevated. And they can see that when they depolarize the neuron using potassium, that the calcium change that's induced by potassium application is smaller in the neurons that have been treated with cisplatin. And so they're showing you that, mitochondria are affected, and calcium is affected. Calcium signaling is affected, and calcium reuptake, calcium dynamics are all affected. So calcium dynamics are slower, blah, 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 blah. And how they do this and what the methodology that they use to do this is not really clear to me exactly how effective it is, but because it's not my field. But here they show that they have these they can look at mitochondrial dynamics. I'm not really sure how they do it, but in this figure that's what they show. So figure 3, they go on to show with fluorophores that mitochondria are actually moving from the astrocytes to the damaged neurons, and this is a great figure, a really great figure, because this is really the bomb. I mean, I don't think anyone ever suspected this. I don't think anyone's known this. The idea that mitochondria can move between cells, I don't even know if that's known in biology. I certainly don't know if anyone knows that astrocytes can donate mitochondria to neurons. This is an extraordinary result already. But now, they're just going to go a little bit farther to be sure that you believe them, and do a couple controls where they're going to try and mess with genes associated with mitochondria in astrocytes to see if they can reverse these effects or tweak them somehow, to give a better idea that it's really mitochondria that are doing this. And so far they've shown you that cisplatin can change neuronal survival, that it can change neuronal calcium dynamics, and by co-culturing neurons with astrocytes, the effect of cisplatin is normalized. So what they do is that they first mess with a GTPase, which is involved in mitochondrial movement and mobility. And when they mess with this GTPase, it reduces the effect seen in the astrocyte co-cultures. So by messing with the GTPase, and I think the GTPase has something to do with mitochondria and mitochondrial regulation, or mitochondrial calcium signaling, or something like that. I'm not really sure. But they're doing something molecular to confirm that it's really the mitochondria moving from the astrocyte to the neuron that causes the calcium signaling to normalize. That's the idea here. I gotta be watching these cars. There's way too many people in them. Sorry, I can't talk right now. Almost there. So that was figure 4, where they mess with the GTPase and they show that the effects are dependent. This GTPase can alter the effects. Figure 5 shows the restoration of calcium dynamics, very specifically in the cells that get astrocytes, or to get astrocytes mitochondria. No, sorry, figure 5 is the rescue of calcium dynamics, and the neurons are treated with a potassium depolarization in order to see and measure calcium responses. And they show that those calcium responses are restored. So the first figure where they're looking at responses was actually looking at the signaling in mitochondria and showed that their resting membrane potential returned to normal. And now in figure, I believe it's figure 4, they're looking at the Ndidi-Ese and showing a change. And then in figure 5, they actually show that the calcium dynamics in the neurons themselves are rescued. So that's really cool because now you have this prep. It's a neuron prep. If you co-culture with astrocytes, the chemotherapeutic agent doesn't really have any effect on neuronal function, although it does if you don't have the astrocytes around. If they mess with the mobility of mitochondria, then you see that the effect cannot happen. And if they look at the calcium signals in the neurons themselves, those calcium dynamics are restored in neurons where when the mitochondria are from the astrocytes, and they can do that with M-cherry labels. Nice stop. So figure 6 is going to look specifically at whether or not neurons got mitochondria or not, and how many mitochondria they took up, and then how their calcium signals correspond to that. And what it shows is that the ones that get the mitochondria are the ones that have the best recovery from the cisplatin treatment. So I think this paper is really cool because it does show that mitochondria can move from astrocytes to neurons. And the question is, do they do this normally? Is it a replenishing process? Is it something that only occurs during damage? Or is it something that really happens as a homeostatic mechanism? These are the kinds of questions that become really interesting because, again, if it is homeostatic, then they essentially, in this paper, have discovered a new mechanism of homeostatic plasticity in the brain. I mean, mitochondria have their own DNA. Mitochondria are responsible for a lot of calcium control, a lot of calcium dynamics. They're responsible for the energy of the cell. So the idea that astrocytes can contribute their mitochondria to neurons, I mean, even if you want to argue that this paper is really doing it in an artificial way, artificial system, cultures, and using a poison, it has resulted in a spectacular observation. And from a biological perspective, I'm just not really sure when this could have been seen before. Jesus. There's some ice on the bridge. I mean, this week has been great. First, a paper shows that some synapses are responsible for learning in the PFC twice. And now we get this myelin paper that shows that myelination is responsible for learning adjustments and learning plasticity. And then finally, same week, we get evidence that mitochondria can transfer from astrocytes to neurons. And so the relationship between neurons and astrocytes has just changed drastically if they can replace their energy factories. if mitochondria can be renewed by astrocytes. Maybe they even are renewed by astrocytes, like as a mechanism. And if that's the case, then holy shite balls, they've uncovered something really cool. Because that's a place, of course, where we haven't been looking for effects of in neuropsychiatric disorders. We're not looking for aberrant transfer of mitochondria from astrocytes to neurons as a mechanism by which neuropsychiatric disorders could be generated. But if this observation is true, then it's clearly possible. And it has as many implications as the paper from Paul Franklin's lab showing that myelination can be more or less responsible for learning. Because again, you put these processes into a context that heretofore they have not been in. Nobody has shown the effects of myelination on learning so well as that paper, and no one has shown the effects of the fact that astrocytes can donate mitochondria like this. And given the fact that we know that mitochondrial disorders are a thing, then it's very interesting to think about this ecosystem and whether neurons can just ask for more, or whether neuronal activity drives this response, or I mean, it all becomes very interesting from this perspective that we just don't know, and we didn't know, and since we didn't know, having to factor in such a new piece of information, it's just it's a big adjustment. It's not a small adjustment in terms of how to think about how the brain works. It's a pretty big adjustment. I mean, let's go all the way to the far end of this one and think, what if astrocytes are responsible for the generation of mitochondria and the supply of mitochondria that neurons actually burn through all the time in normal life? And so then you just have this constant replenishment from the astrocyte pool of mitochondria in neurons. That would be a major discovery. And I would argue quite unexpected. I think mitochondrial regulation is one of those stories in biology that it will end up being more and more and more important as we get to know them. The problem is they're too small to study, very difficult to manipulate, and small manipulations have huge effects, so oftentimes just deadly. Joining me on my ride. That was a paper by the Heijnen lab. The first author is English, and this paper looked at a cancer biology drug, a chemotherapy drug, and looked at how it affected neuronal cultures. Specifically, what they show is that neuronal survival and neuronal calcium signals are affected by this cisplatin drug. And what they show is that co-culturing with astrocytes can normalize the calcium signaling and increase neuronal survival in these cultures. Interestingly enough, what they also show with some fluorescent tags and some messing with genes associated with mitochondrial trafficking, they show that mitochondria are transferred to the neurons, and that the transfer of these mitochondria is what normalizes the calcium signals in these preparations that are treated with this cancer drug. What this shows is that very specifically mitochondria can be donated from astrocytes to neurons, at least under stress. And now the real question becomes, is this something that happens normally? And if so, to what extent does it happen? Why does it happen? What kinds of effects can it have? Because we know that neuronal function can be affected by mitochondrial dysfunction in a wide variety of in a wide variety of studies and a wide variety of association studies with genes associated with mitochondria. We know that mitochondria are important. And this is the kind of the first, I think, huge paper to show, or a result to show, that mitochondria can be affected, how they are affected, and then how they could be replaced. So imagine a scenario where neurons actually use up mitochondria and are constantly having their mitochondria replenished by astrocytes. If that's the case, then there's a whole nother site of plasticity and another site of therapeutics and another site of possible source of these neuropsychiatric disorders. So potentially a really exciting paper right now just in neuronal cultures, but showing something that hasn't been seen before. Merry Christmas, Happy New Year. I'll see you around the break, and after the break I'm going to try and come back with 3 a week. Thanks for joining me in 2019, and I hope to see you next year. Bye-bye. Stop.